Hello, I'm Evan Ehrenberg. I'm a second-year graduate student here at MIT in the Brain and Cognitive Sciences department. I work in the Sinha lab, where I research face identification. I take a very interdisciplinary approach towards understanding and replicating face identification called computational neuroscience. This field of science is very new and has only been developed in the last decade or so. It combines neuroscience with math, computer science, and psychology in an attempt to better understand the computations the brain is performing, and how similar computations can be performed by artificial systems, like computers, to produce the same behavior, again in this case recognizing a face. During this video, I hope to introduce you to this developing field, show you some cutting-edge technology for brain imaging, and walk you through an experiment that I'm currently developing that will tell us how the brain represents faces. But first, why should we study artificial intelligence? What does it offer society as a field of research? When you think of AI, you might go straight for the science fiction version. The Terminator might come to mind, as could Sunny, Johnny 5, or WALL-E. You might even think of some non-robotic AIs, such as HAL from 2001: A Space Odyssey, or the agents and other programs in the Matrix. While there are many humanoid robots under development, the majority of AI today is not embodied in living tissue over metal endoskeleton, and instead resides in our computers, much like HAL or Agent Smith. Unlike HAL, today's artificial intelligence has not achieved consciousness, and is not nearly as intelligent. The intelligence displayed by HAL is what we call strong artificial intelligence. Strong AI is defined as an artificial intelligence that is comparable to human intelligence, and is sort of this big goal way off in the distance that everyone in the field wants to reach. When strong AI is achieved, it's hypothesized that science will make more breakthroughs in a week than all the scientific breakthroughs in human history combined. Now, the other type of AI is weak AI, and is what primarily exists today. Weak AI is any program that behaves in an intelligent manner, something that adapts to the data it's provided with. A lot of the brain's computations can be thought of as AI because so much processing goes on before we're made consciously aware of the outside world. The brain is constantly organizing the senses so that prefrontal cortex, the area responsible for higher level cognition and the seat of consciousness, can access just what it needs to make decisions quickly and easily. Now, you might be disappointed to know that strong AI won't be here for some time, but weak AI is extremely useful. In fact, it's already all around you, and it's getting better every day. GPS units use AI to determine the most efficient route for you to get home, and cars equipped with lane departure warning systems use it to ensure you don't drift out of your lane as you drive along this route. It's used in Photoshop to replace that photobomber with a realistic, synthesized background. Netflix uses AI to suggest movies to you, and iTunes to suggest songs. Okay, so AI is definitely worth researching. But why is it such a hard problem? And why should we study the brain to engineer it? To answer this, I'm going to show you just how powerful and complex your brain is. This organ in our skull does so much more than your math homework. For example, the amount of processing that goes into something as simple as picking up a jug of milk can often be surprising. Try this experiment. Find your textbook and hold it in your outstretched hand. Place a heavy object on it, like another textbook. Now, quickly lift the item off of the book. You'll notice that the bottom book doesn't move when you do this, or when you place the item back down. Your brain automatically calculates how much the top book weighed in your left hand, and subtracted that from the weight in your right hand to determine how much the muscles in your arm should contract to keep the new weight at the same height. Now have your friend lift the object off the textbook for you. In this case, you no longer know when the item will be removed, or how much it weighs, and your arm shoots up for a second because your brain doesn't know how to adjust the lifting algorithm to account for the lesser weight. Similarly, when they replace the book, your arm falls for the same reasons. Human-like movements such as these has been a big goal and a big problem in robotics. Honda has created Osimo, one of the most advanced bipedal humanoid robots. Now, Osimo only has to control 34 motors compared to our body's 639 skeletal muscles, but the problem of planning proper body and limb movements is still an extremely difficult one. Let's watch a video of Osimo walking upstairs. So you can see it started out pretty good, albeit slowly. But the moment it encountered a problem, it couldn't adapt its movement to prevent itself from falling. Let's take a quick look at some human locomotion. Humans and other animals are constantly adapting their movement as the world around them changes, and we do it very fast and automatically. If we didn't, parkour wouldn't be a sport, and humans would have died out long ago. Let's look at something that humans find even easier than walking or grasping: seeing. Well, what do we do to see? We just open our eyes and we see, right? I mean, we can't decide not to see, it just happens. There's absolutely no conscious effort involved. It sounds like a pretty simple problem for AI to conquer, and this is exactly what the pioneers of AI thought when they started the field in the summer of 1956. Why can't we just plug a camera into our computer and have it see? The reason vision is such a simple feat for us is because 30% of our cerebral cortex is devoted to processing it. Around 5 billion neurons are constantly making sense of the input from our eyes, so we don't have to. By the time the prefrontal cortex gets the data that started out in our retinas, the image is all sorted out and nicely labeled for us. If we could plug a camera into visual cortex and then have that feed into a computer, it would see just as easily as we do. Our brain does lots of extremely complex computations for us subconsciously, so that we can focus on what matters, the higher level objects around us that are represented in the sensory data that we receive. The more of our brain devoted to a task, the easier it is for us to do. Vision is an extremely difficult problem to solve, but because so much of our brains are optimized to solve it, it seems easy. Other tasks, like multiplying 2 3-digit numbers, can seem difficult, even though they're actually very simple computational problems. The reason we find them to be so difficult is that there is no area of the brain that's processing the numbers for you. It's all having to run at a very abstract level in your prefrontal cortex. Since there was no evolutionary pressure for our brains to do math quickly, we never evolved cortical areas that are hardwired for it. Computers, on the other hand, are hardwired to handle math calculations, and therefore perform them quickly and efficiently. And now you can see why it's so difficult to develop artificial systems for vision, audition, and motor coordination, because these are all very difficult problems, and we aren't applying a lot of optimized computations for pre-processing of it, because we're still learning how. We're learning from the brain. High-resolution vision is such a difficult problem to solve that even our brain cannot learn to see in high resolution all at once, so we've evolved a way to develop it slowly. We are born with 2400 vision, specifically so that we can learn to see. To put that in perspective, people are considered legally blind past 2200 vision. If the newborn brain received all the data we can get from 20/20 acuity eyes, it would be overwhelmed with too much data to pick out the underlying patterns. When we start with 2400 vision, our brains can parse the visual input and begin to learn how to see. As our brains learn some of the basic visual features, our eyes gradually gain acuity until they reach 20/20 vision around 6 years of age. And the brain is able to keep up because by this time, it's already learned some of the basics of vision. I like to think of the brain as my cheat sheet for how to engineer vision in a computer. I have this complex system that does exactly what I want my computer to do, so I study neuroscience to find out what our brains are doing, so I can design a computer program to do the same thing. So, without further ado, let's take a quick look at some basic features of the brain and neurons. The neuron is a very complex computational cell, so we'll just look at some of the basics here. Neurons are composed of 3 main parts: the soma, dendrites, and an axon. The soma is the cell body and doesn't differ much from other cells. Dendrites and axons are unique to neurons and allow neurons to compute. The axon is a large branch that lets the neuron communicate with other neurons. The axon ends with several synapses which make connections with the dendrites of other neurons. When a neuron is activated, it fires an electrical action potential down its axon. Here we have a blown up version of one of these synapses. When the action potential reaches a synapse, it activates a chain of events which fuse these vesicles with the cell wall, dumping neurotransmitter to the postsynaptic cell's dendrite, where the neurotransmitters activate these receptor sites right here. These receptors will come and go over time, and allow the neurons to change how inhibitory or excitatory they are towards each other. Neuroscientists call these values weights, and they vary from cell to cell. Changing the weights between neurons is essential for learning new things. We call this ability to change weights and rewire connections neural plasticity. Our brains are most plastic when we're very young, before about 6 years of age. Our brains are still solidifying until about 24 years of age. After that, they're much less plastic than before. Of course, you can still learn new things when you're older, but there are some things you can't do, like rewire your entire brain. You might think that you wouldn't survive removing half of your brain, but when you're very young, again about 5 years or younger, your brain can rewire itself and fix almost any damage it encounters. There have been children with very severe epilepsy who have hemispherectomies, where a neurosurgeon removes the epileptic hemisphere of the brain. These patients recover within a year, and years later, you still can't tell anything's wrong with them. But if you scan their brain, you'll find that half of it is missing. Now that we know a bit about neurons, let's go to the wet lab and talk about how many there are. Alright, so now I have an actual human brain here that's been partially dissected. Your brain has 85 billion neurons, and about 70 billion of them are in this little brain down here, called the cerebellum, which actually means little brain in Latin. The other 15 billion are found in the rest of the brain, which we call a telencephalon, or the cerebrum. If we take a cross-section of the brain, we can see that there is actually white and gray matter. The white matter is composed primarily of axons and gets its white pigmentation from myelin, which is a fatty tube that insulates axons, which communicate over long distances. Myelin speeds up the action potential and ensures that it doesn't die out along these long axons. Gray matter is where most of the neurons' bodies and dendrites reside and is called cortex. These wrinkles in the brain are called sulci and gyri. The sulci are any of these valleys, and the gyri are any of the hilltops that you can see from the outside of the brain. Gray matter in the cerebrum is called neocortex and does most of the computations in the brain. Neocortex is composed of 6 layers of cells and can learn just about anything. The neocortex has about the same thickness and area as this cloth napkin. Because neocortex is such a great computational medium, we want to fit as much of it in our skulls as we can, so we scrunch it up. And that's why our brains appear wrinkly, because we're maximizing the area of neocortex we can fit in the volume of our brains. Crumpling neocortex in this way also allows for faster communication, because now a neuron over here can communicate with one right here, whereas before it had to send an action potential all the way across the brain. Monkeys, cats, and rats have correspondingly less of these wrinkles, and consequently have less neocortex for the size of their brain, and are therefore less intelligent. You'll remember that I said 70 billion of our 85 billion neurons are in the cerebellum. You might be thinking this is a super important area of the brain, but actually it's not. It just has a very complicated job. The cerebellum performs motor coordination for us. It makes sure that all of our movements are smooth and fluid. When we look at the cerebellum, we see one very prominent feature, and that's that it is much more wrinkled than the cerebrum. In fact, if you look at a slice of it, you can see just how wrinkled it is. One reason so many neurons are in the cerebellum is because it fits so much cerebellar cortex in such a small area. The other reason is that the neurons found here are much smaller than those found elsewhere in the brain. These neurons are called granule cells and are the most common neuron in the brain, even though they reside almost exclusively in the cerebellum. Now let's look at the big picture, the lobes of the brain. The cerebrum is divided up into 4 lobes, and they are the frontal lobe, the parietal lobe, the occipital lobe, and the temporal lobe. The frontal lobe is perhaps the most important for humans, for it is where high-level cognition and muscle movements are processed. The very front of the frontal lobe is called the prefrontal cortex and performs the high-level cognition, while the back of the frontal lobe is where we find the motor strip, which performs movement. Just behind the motor strip, we find the somatosensory strip, which resides in the parietal lobe. The parietal lobe is known for integrating all of our senses and preparing them to send to the frontal lobe for decision-making. Just behind the parietal lobe is the occipital lobe, which we'll talk about a lot in this video because it is where vision is processed. Finally, we have the temporal lobe, which also does some visual processing, but is perhaps best known for learning and memory, because this is where the hippocampus is. Here I've taken out the right temporal lobe of the brain. The hippocampus is this seahorse-shaped object right here. This takes in sensory input and stores it in the neocortex of the brain. It also aids in retrieving these memories, though they can be accessed by other means too. It's for this reason that people with damage to hippocampus can't form new memories. Patient HM is a very famous example of this. He had part of his temporal lobe removed to cure his epilepsy. The surgery removed most of the hippocampus, and from then on, he could only remember events which took place before the surgery. Interestingly enough, he could still be conditioned to fear something, like a person who constantly greeted him with a gag hand buzzer, but he would never know why he felt afraid of them. This suggests that simple conditional learning does not require the hippocampus. The 2 hemispheres of the brain communicate via a small cluster of axons called the corpus callosum, which you can see here. It's just this small area of white matter. Axons from the corpus callosum project to the neocortex via the corona radiata, which is this sort of radiating crown. These are the axons which come from the corpus callosum and project up to neocortex. Neocortex also projects back to the corpus callosum via the corona radiata. Finally, I just want to clarify 2 common misconceptions about the brain. First, we don't use 10% of our brain. We use all of it. As I demonstrated with hemispherectomy patients, We can still function fine with 50% of our brains, but if we have all 100% intact, we'll use all of it. Second, movies often portray neurons as floating in space with branching axons and dendrites in between. In reality, however, neurons are very tightly packed with no space between them. Here's an example of a rainbow stained slice of cortex, showing just how packed in they are. These neurons are dyed different colors, so they'll show up individually. It's really quite a beautiful sight. Let's not stop here, though. I think you know enough about the brain to take a closer look at vision now. Where else should we begin other than the eyes, where vision starts? You've probably already heard about how the eye's lens focuses light onto your retina, and how the image gets flipped upside down when it's focused. And if you didn't, you know now. What we're interested in, though, is the computational aspect of vision, so we're going to skip the lens and get right to the retina. You may not know, but there's actually computation going on in your eye right now as you watch this video. You see, after light activates a photoreceptor cell in your retina, there is a short series of other cells which perform pre-processing on the image before the signal gets sent out of the eye via the optic nerve. This pre-processing sets up the building blocks for edge detection later in the brain. The final layer of cells in the retina, called retinal ganglion cells, give a very specific response to light. They have what are called center surround fields, which means they treat light in the center of the receptive fields differently than they treat light in the periphery of the receptive fields. There are two main types of these cells, on-center and off-center, and they do exactly what you'd think they do. An on-center cell responds best when light is shown in the center and nowhere else. They don't respond much when there's light over the entire receptive field or when there's no light at all. And they're absolutely silent when the surround is activated and the center has no light. Off-center cells behave in exactly the opposite fashion. Later, neurons in visual cortex will take this information to create edge detectors. You can see, when we line up these center surround fields and have them send their input to a single neuron, this neuron will respond best to lines at this orientation, because it's going through the center of all the center surround fields and very little of the surround. Versus, say, a line at this orientation, we're only going through the center of one cell. Now, what's really interesting is that not only does this cell like this one orientation, but it likes it only in this position. You see, if I draw a line at the same orientation, but in a different position, it just goes over the surrounds of the cells. It doesn't go through any of the centers. And so this cell isn't going to be very active at all. It's a simple system, but it's a great one to look at to understand how neurons work together to create complex output. Let's continue on down the optic nerve and into the brain. You may be aware that the right side of the brain processes input from the left side of the body, and that similarly, the left side processes input from the right side of the body. This is true for vision as well, but instead of the right side of the brain taking input from just the left eye, the right side of the brain actually takes in input from anything on the left side of the visual field. To do this, the right brain hemisphere actually takes input from the right side of both eyes. If you assume these blue and red circles represent distinct objects, you can see how the light that bounces off of them ends up on the opposite side of both eyes. So light bouncing off of this red object, which is on the right side of your visual field, goes to the left side of both eyes. Similarly for the blue object. In order to have depth perception, each hemisphere takes input from one side of both eyes. This crossing of the optic nerves is known as the optic chiasm and is right here. Past the optic chiasm, the optic nerve passes through the lateral geniculate nucleus, or LGN, for some further processing before it finally reaches the primary visual cortex, or V1, all the way in the back of the brain, in the occipital lobe. Primary visual cortex, otherwise known as V1, is composed of cortical columns of neurons. These neurons are lined up to respond to different orientations, so angles of lines. So V1 is still doing some pretty simple processing, and V2 does mostly the same thing at a larger scale. So each neuron is taking input from a larger area of the visual field. After V2, visual cortex starts to branch off into two streams, the ventral stream and the dorsal stream. Dorsal refers to the top of the brain, like a dorsal fin on a dolphin, and the ventral stream refers to the bottom of the brain. V3 and V5, we take the dorsal path up here. V5 is responsible for detecting motion in the visual field, and tracking and predicting where objects are moving. Since knowing how the world is moving is so important for planning your own motion, output from V5 is used extensively for adapting motor control, and this is expressed in V5's close proximity to motor cortex, which is right about here. V5 is also referred to as area MT, or middle temporal cortex, so just keep that in mind. The other path I mentioned, the ventral stream, is where we'll find V4. The ventral stream is where object identification takes place. V4 sends its output to IT. IT stands for inferotemporal cortex and just tells us where it is, on the bottom of the temporal lobe. It's in IT that we finally start to recognize the objects in our environment, and where we find something called the fusiform face area, or FFA, which is very important for my research. It's called the fusiform face area because it lies in the fusiform gyrus on the bottom of the temporal lobe. FFA is one of the few areas of the brain that shows a specific response to an individual type of visual stimuli, a face. Even though we know other types of visual recognition are taking place in the ventral stream, we haven't found areas that show a recordable response toward just a few stimuli and not others that is as strong as what we find in FFA. Because FFA is still working at an unconscious level in our brains, its responses are not identical with what we think of as faces. While FFA shows the strongest response to faces, it will respond to like a headless body as well, something that we wouldn't qualify a face. Considering the current level of AI, the responses from fusiform face area are a great place to look for hints as to what features we use to classify objects. Now when I say features, we can think of these as something like the size of our ears or nose, but in reality, the features the brain and AI software make use of are more like the relative distance between the eyes with regard to the length of the nose. Anyway, we can manipulate individual features in a face, display these to humans positioned in a brain scanner, and observe how these morphed faces modulate response from inferotemporal cortex, and fusiform face area specifically. If we manipulate a feature and FFA doesn't show a change in response, we know that this is not a feature that the brain is sensitive to. If a feature, on the other hand, were to cause a fluctuation in FFA response, we know that this is a feature that the brain is using for face detection, and we should represent this as an important feature in our computational model or AI software for face identification. There's plenty of brain functions to study, and a wide variety of ways to study them. Neuroscience has just recently gained some important brain imaging tools that have greatly impacted the field and how science is performed. fMRI, EEG, MEG, and PET imaging provide scientists with a non-invasive way to view the brain's structure and record neural responses by measuring blood flow and electrical and magnetic fields produced by the brain. I use EEG and MEG in my research of face identification. EEG stands for electroencephalography, and MEG stands for magnetoencephalography. Now let's break these scary words down. So electro refers to the electrical fields that this device measures, and encephalo is Latin for brain. And graph means to measure, so EEG is a device which measures the electrical fields of the brain. MEG similarly means a device which measures the magnetic fields of the brain. These devices are essentially measuring two dimensions of the same thing, since everywhere an electrical field is moving, a magnetic field is generated too. Magnetic fields pass through the skull more easily than electrical fields, which allow us to record from neurons at a higher frequency with MEG than with EEG. Unfortunately, because of the way magnetic fields are generated, we can only detect them from the neurons in the sulci, or valleys of the brain. To detect neurons residing in the gyri, or the surface of the brain, we have to use EEG, which detects both neural activity from the sulci and gyri. Thankfully, EEG does not interfere with MEG, and so scientists such as myself will often place an EEG cap, such as this one, on a subject when they use MEG in order to pick up these additional signals. While this EEG cap is fairly small, the MEG machine takes up an entire room. This is because the magnetic fields, while they carry more information through the skull, are much fainter than the electrical fields produced by action potentials. Let's take a look at the MEG facility. Alright, so this room on my right is where the MEG is housed. Over here, you can see the liquid helium that we use to cool the sensors to 2 degrees Kelvin in order for them to detect the faint magnetic fields that are produced by the brain. Over here, you can see all of the computers that we use to control the stimuli we give our participants. The MEG itself is housed in a magnetically sealed room. You can see there are 3 layers of pure aluminum bound to ferromagnetic layers. This makes sure that none of the magnetic fields out here interfere with the MEG's ability to record from our participants. Alright, so to enter the room, I've had to remove all metal from my body, including my glasses and my mic. So I apologize if the audio quality goes down a bit for the next few seconds. But here, you can see the MEG. It's quite a big machine. Just on the inside of this rim are over 300 SQUIDs, or superconducting quantum interference devices. And these are the sensors that the MEG uses to record from your brain. Now, if you were a participant in one of our studies, you get to sit in this nice, comfy leather chair, do a few simple experiments, and that's it. I should mention that the MEG is completely non-invasive, so nothing goes in your brain. It's just like sitting in the hairdryer, the hair salon. One psychophysics experiment, which I'm currently working on, will utilize both MEG and EEG, and will focus on determining IT's ability to differentiate familiar faces from those unfamiliar to us, and even to distinguish individual familiar faces from other familiar faces. MEG and EEG provide excellent temporal resolution, meaning that they can tell us exactly when groups of neurons in the brain are firing. However, it has relatively poor spatial resolution compared to other brain imaging techniques such as magnetic resonance imaging. It's for this reason that I'm looking across all of inferotemporal cortex for data indicating that the brain is making these distinctions, because FFA is so small that we don't get much data from it using MEG. For my database of familiar and unfamiliar images, I'll be showing my subjects. I've chosen 200 images of American celebrities, composed of 4 celebrities with 5 different photos of each. I've also included a similar 200 images of foreign celebrities, which are unfamiliar to my American subjects. Subjects will be given a series of tasks, ranging from passive viewing, where there is no task, to simple change detection, where now and then a blue dot will appear on the screen instead of a face, and participants will have to push a button to indicate that this has happened. These tasks are actually called distractor or dummy tasks. We don't actually care about their performance on these tasks beyond making sure they're paying attention and not daydreaming. In fact, we want to make sure that these tasks are ones that won't interfere with the processing of the faces. However, we will be running a control during the experiment where participants are asked to press a button if they recognize a face. By having subjects engage in this task we are measuring, we can see how top-down processing and attention affects IT's response to familiar and unfamiliar faces. To determine if the brain response is responding to the identity of the face versus some set of visual features, we will also flash the names of the celebrities on screen. If we find a similar response for the names as we did for the faces, then it looks like IT is doing some very high-level recognition of the person and can be activated by their face or their name. What we expect to find, however, is that IT and FFA will show a preference for the faces and not the names, meaning that they are face detectors and not identity recognizers. Once we've gathered data from several experiments and have done our data analysis, we can start to apply our newfound knowledge towards computational models of the brain and AI. While computational neuroscience is a novel field, you can see that it is a very useful one. The advances it makes not only help to engineer AI, but also broaden our knowledge of the brain, aiding in the medical treatment of brain diseases and trauma. Beyond being a great field for advancing technology and medicine, computational neuroscience is a fun field to work in. Because it's so interdisciplinary, you can work with humans, animals, brains, computer programs, and even robotic systems. There's something in it for everyone. I hope you enjoyed this video, and that I've inspired you to learn a bit more about the brain and artificial intelligence.